Good morning, everyone. Good morning. Good morning, everyone. Good morning. Good morning. If anyone misses human physiology classes, please kindly watch the recorded. Don't miss any class at all. Good morning. Good morning, everyone. So, friends, we were doing about the membrane transport in plants. There's a little part is left. We will finish it off. Okay. So, friends, try to understand. We were discussing about transport mechanism. In the yesterday class, friends, we completely understand about water channels, water carriers, water pumps. We understand, and later we started studying about the transport of water as well as other ions and molecules through the apoplast and symplastic pathway. So I hope every student remember. Good morning everyone. So I told you very clearly, the moment you heard the name of a apoplast, what is the common statement that I want you to remember, friends? Apoplastic pathway means it is always via intercellular space, right? Of course, friends, the moment you heard the name of a symplastic, what is the most common statement that I want you to remember, friends? Symplastic pathway, it is exclusively via what, friends? Plasmodesmata. I hope every one of you remembered everything in the yesterday whatever we discussed. So this is the last part we discussed, right? So friends, we discussed that xylem consists of a parenchyma, trachea elements. I told to all of you, wherever you read the parenchyma, remember only one common point. The parenchyma is the living tissue. Then friends, the trachea elements are the dead tissue. And I told you, friends, you should not think that since parenchyma is the living tissue, so you should not think that the water movement majorly occur through the parenchyma, which is absolutely wrong statement. The water movement always occur via trachea elements, because friends, they are dead cell. Dead cell do not contain anything inside. Hollow lumen, a great space is present. So through that space, all the water molecules can easily flow. So friends, this particular trachea elements are broadly categorized into tracheates and vessel elements. Kindly don't forget. And I hope you all remember. Friends, among the vessel elements and the tracheates, which can show the lower efficiency in water conduction, obviously friends, tracheates will show the lower efficiency in water conduction, whereas friends, what about the vessel elements? Vessel elements always shows the higher efficiency in the water conduction. Please kindly don't forget it. And if you all quickly recall the differences, what did I tell to all of you? Tracheates are long. What, friends? Tracheates are long, elongated, and most importantly, friends, they are spindle fiber in shape. Don't forget it. And you all people can see the picture clearly. They are having a pits or pores. What did I tell to all of you? Through the help of pits and pores, they will be responsible for the transport of water. But friends, when you look into the this particular vessels, what we discussed, vessels are not elongated, vessels are shorter. Friends, even though vessels are shorter, but vessels are wider. And along with the pits and pores, what is the speciality of the vessels, friends? The speciality of the vessels is they are having perforated plates. So friends, with the help of that particular perforated plates, the water conduction will become more efficient. Vessel means what? Angio. Angio means vessel-like structures. So the vessel elements are abundantly present in the angiosperms, which are highly developed plants. That's why we can say vessel elements are having the higher efficiency in the water conduction. Kindly don't forget that. Vessel elements, tracheates. Okay, finally where the water reached, friends? Everyone look into the picture carefully, all of you. Finally water reached where? Water reached to the root. Okay, from the root where the water has to flow, friends, the water has to reach the 100 feet tall plant. Now every student, please listen. How does this water molecules are reaching this 100 feet tall plant? Everyone pay attention. With the help of cohesion and with the help of adhesion, and friends, most importantly, the force or the pressure which is generally created by the transpiration pull. Kindly don't forget this. So all of you, please try to understand. I told you, right, which elements are the better conducting? Tracheary elements. I want every student has to look into this diagram, friends. You all people can see, no friends, here one water molecule can be associated with the another water molecule by the process of cohesion. How, friends? You can see here this is one water molecule, this is the another water molecule, correct na? So two water molecules are connected with each other by OH linkage. Like that, all the water molecule can make one chain, right? Then friends, how does the water molecule attaches to the xylem? The water molecules attaches to the wall of the xylem by the process of adhesion. One is cohesion, another one is the adhesion. And friends, the most important one you people has to remember for the exam. Okay, water molecules are attached with each other, that's correct. Water molecules also attaches to the wall of the xylem, that is also correct. But the question is here, how does this particular water chain moving up to the 100 feet tall plant from the root? So there should be some pressure, there should be some force. So what is that particular force or pressure? We can call it transpiration pull. See friends, when I use a word transpiration pull, you have to know it very depth, how exactly the movement is going to takes place. I will explain elaboratively how exactly the movement occur. I want you to pay attention. I will explain nicely this. Just focus. Okay, all of you look into this. Just focus whatever I am drawing. Okay, so this is the leaf. Okay, let's take the cross section of leaf. Just try to understand what I am saying. You have to know this concept. Okay, this is the upper epidermis. Okay, then friends, followed by the lower epidermis. And we know very well the lower epidermis contain guard cells, which contain what, friends? Stomata. Guard cells which contain stomata. And friends, all the students listen to me carefully. Here what kind of cells are there, friends? Palisade mesophyll. Friends, whenever I use a word palisade mesophyll, this is also asked a question in the exam, friends. Palisade mesophyll, friends, they are associated very tightly with each other. And friends, palisade mesophyll do not have any kind of the intercellular spaces because they are very tightly associated with each other. Then after the palisade cell, what kind of mesophyll cells are present? Spongy mesophyll cell. You all people can see, friends. Spongy mesophyll cells are arranged very freely, and moreover, friends, they are having a great intercellular spaces. Now let me tell you, what is our goal, what we have to understand. This is what we have to understand. This is the water chain. How does this particular water chain is coming from the root to the tip of the plant, very tall plant. All the students listen. Whenever there will be a loss of, whenever there is an opening, whenever there is an opening of this particular stomata, imagine all these red color dots are the water molecules, friends. All are water molecules. The moment, listen to me carefully, the moment stomata get opened, all the water molecules go out, is it? Now every student listen. When the water molecules are going out, understand everyone, between the spongy mesophyll cell there is a lot of gap. Between the spongy mesophyll cells there will be a lot of gap is present, correct na friends? Through that gap, you know what the spongy mesophyll cells will do? They start pulling the water. Imagine the concept, whatever I am saying. There is a lot of gap present between one spongy mesophyll cell to the another spongy mesophyll cell. Through that gap, what the spongy mesophyll cells will do? They start pulling the water. Friends, understand, when they start pulling the water, immediately you know what happen? It create a pressure. It create a pressure on this water chain. As a result, friends, this particular water chain, you all people can see here, this water chain is coming up. Here also you can see, my friends, whenever all the students listen, here stomata got opened. Upon opening of the stomata, all the water molecules are losing. As a result, what happened? In the picture you can see very clearly, these are all palisade mesophyll. You can see, friends, there is a no intercellular spaces, and these are all what? Spongy mesophyll cell. There is a great intercellular spaces. What they are doing, friends? They are pulling the water from the intercellular spaces, which create a pressure on this particular water chain. That is how water chain is coming from the root to the tip of the shoot. Very, 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 very simple point it is. That is how, friends, water molecules moves up. Okay, now friends, where does the water molecule reach? Water molecule reached to the tip of the plant. Say an example, it reached the leaf. Now what happens in the leaf? Now leaf will take the water, leaf will take the carbon dioxide from the atmosphere. What the leaf will do? With the help of sunlight and chlorophyll, it will convert the carbon into glucose. But every student listen to me carefully. Where does the sugar synthesized? In the mesophyll. Absolutely true point. Where the sugars are synthesized? In the mesophyll. But the point is very clear. Only sugars are required for leaves. No, right? Friends, sugar synthesized in the leaf, but every part of the plant body, root requires sugar, shoot requires sugar, tubers require sugar. Every part of the plant body, friends, require the sugar compulsory. There is no doubt about it. Now the point is here, the sugars are synthesized in the mesophyll cells of the leaf. From the mesophyll cells of the leaf, how does the sugar transported to the all other parts of the body? So it will be transported via phloem. How it will be transported, friends? Via phloem. That's what we can call the translocation in phloem. Translocation means what? Movement. Movement in the phloem. That is what exactly we can call the translocation. Friends, every student keep it in your mind. Wherever the sugars are synthesized, that area we can call the source. synthesis occur. Wherever sugars are not present, we will be calling that particular area as the sink, no sugars. Now everyone remember, from where to where these are moving, friends? Sugars are always move from the source to the sink. So the photosynthetes means the solutes which we produce. What we will produce? Of course, we will produce the sugar. So the sugars always move from the source to the sink via phloem sieve tube elements. Now once again, I want you people to concentrate here. Friends, we discussed that xylem contains xylem parenchyma, xylem contains tracheary elements. That is what we people study. Now all of you listen, same way I want you to understand, friends, phloem also consists of parenchyma. There is no doubt about it. Phloem also consists of a parenchyma, and friends, phloem can also consist of the sieve tube elements. Kindly don't forget. See friends, the sieve elements of the phloem, friends, this is more important than the xylem. I am not saying that questions always comes from the translocation in the phloem. No, questions will come from xylem as well. But friends, most of the time they used to ask specifically from the translocation in the phloem, or in a given 4-marker question they will include the xylem transport and phloem transport, and they will raise the question. So that's why we have to know it very well. Phloem sieve elements, it contains sieve tube elements and it contains sieve cell. And please kindly don't think that parenchyma is not present in the phloem. Parenchyma is present in the xylem also, friends. Parenchyma present in the phloem also. And I already told you, friends, parenchyma is the living tissue. There is no doubt about it. And this is the question already asked in the exam. What is that? Sieve tube element and sieve cells. Please listen the statement whatever I am giving to you correctly, friends. Sieve tube elements and sieve cells they are living at maturity. What, my friends? They are living at a mature stage. Friends, even though they are living at the maturity, they do not have nucleus, right? And they do not have certain other organelles also. So then how they can synthesize the protein? They do not have the nucleus, they do not have the mitochondria. Hence, my friends, they cannot synthesize their own proteins. Now you will get a doubt, sir, for the protein synthesis they are dependent on what? So for the protein structure, phloem sieve tube elements are dependent on a specialized cell. What do you call it, that particular specialized cell? Companion cell. Please kindly don't confuse with this. Once again listen, we read very clearly, tracheary elements are dead in nature. There is no doubt about it. But sieve elements are living at maturity. Even though they are living at maturity, friends, they do not contain nucleus and mitochondria. Hence, friends, they are not involved in the synthesis of proteins. Always remember this. So I want all the students look into this. What are companion cell? All the students listen, because friends, from the companion cell and phloem transport we used to get the questions very oftenly. Companion cells are the one which are involved in the transport of substances in the phloem, requires energy. See, I can tell you one common statement to all of you. Just listen. From this, okay, let me draw here, you will understand very clearly. Okay, look into this, look into this. This is the mesophyll cell. What is mesophyll cell? Where the energy will be, where the sugar will be produced. And in the middle, whatever you can see, friends, that is the companion cell, the green color one. And finally, this blue color one, what is this? This is the, one second. Yeah. This blue color one, this is what, friends? sieve tube element. Now every student try to understand what exactly I am saying. Where does the sugar is synthesized, friends? Sugar is synthesized in the mesophyll. Now you only tell me, does the sugar is basically entering directly from the mesophyll cell into the sieve tube element? Is that possible? Not at all. Every student has to understand. From the mesophyll cell, please focus, friends. From the mesophyll cell, sugars will enter into where? They will enter into the companion cells first. Once again, you have to listen to me. From the companion cell, sugars entered into the sieve tube element. Then they will be transported through the sieve tube element. Finally, they will reach to the zinc cells. Now you will get one question. What is that question? How does this particular companion cell associated with the mesophyll also? How does the companion cell can associated with the sieve tube element also? Because friends, it is linking both, right? I want you people to remember, it is just with the help of plasmodesmata. Don't forget that, friends. What I mean to say, companion cell connected to the mesophyll cell by the plasmodesmata, companion cell connected to the sieve tube element also via plasmodesmata only. Kindly do not forget this particular point. It's a very simple point it is. Okay, so now let me explain you elaboratively. I told you na, when we get a question from the translocation in the phloem, we are going to get 90% question from here itself. That is, how does the movement of sugars will happen? Friends, it's a very easy to understand. Everyone just listen my words. Listen my words. Suppose example, we are shifting a house. Whenever we are shifting a house, what are the steps involved? There are 3 major steps are involved. What are these particular 3 major steps? Everyone listen to me. The first step, sugar start loading. Just imagine house shifting. First we will load all our goods onto the lorry. First step is what? Loading. Loading all our goods onto the lorry. Second step, the lorry will transport from our old house to the new house. So second step is transport. What will be the third step? Of course, once we reach the new house, we will unload everything. Same, just replace the lorry with the sieve tube element of the phloem, replace the goods with the sugar. That's all. So what is the ultimate goal? Ultimate goal is we want to transport the sugars from the source to the sink via sieve tube elements. We all people commonly know that. So friends, what we are doing, first we are loading the sugar from the source into the sieve element. What is the source? We all know very well. Source is the mesophyll. So from the mesophyll cells via companion cell, we will load into the sieve elements. Once we load into the sieve element, what is happening, friends? Movement is occurring through the sieve tube. Inside the sieve tube, sugar has to be transported. Of course, true. I'm going to explain in the next slide. How does that movement occur? That movement basically explained by a mechanism called as Munch hypothesis, based on the name of the discoverer. We can also call it as a bulk flow. We can also call it pressure driven bulk flow. I'll explain you briefly. Finally, once the sugars reach the sink, what happens, friends? Undoubtedly sugars are getting unloading. Simple na? Sugars are unloading from sieve elements into the sink. Such a simple point, friends, it is. Now every one of you listen to me carefully. Try to understand every single point from now onwards. So finally, let's talk about the loading first. So friends, please listen, all of you. Phloem loading also, phloem unloading also. both can be mediated by these two pathways, symplastic pathway of loading, apoplastic pathway of loading. While unloading also, symplastic pathway of unloading, apoplastic pathway of the unloading. Kindly do not forget this point. Now every one of you listen to me carefully. Now you can ask me, sir, listen friends, based on what criteria we say that sugars will be loaded via symplastic? Based on what criteria we can say sugars can be loaded via apoplastic pathway? Based on what criteria? I can tell you very clearly, based on the type of companion cells. Based on what, friends? Of course, based on the type of companion cells. So let's look into the first apoplastic pathway, then we will understand the symplastic pathway. How does the sugars are loading into the apoplastic pathway? Every fellow has to look into the diagram carefully. This is what? Mesophyll. This is what? Companion. This is what? Sieve tube. Sieve tube, companion, mesophyll. Friends, where does the sugar finally entered, friends? Sugar finally entered. This is, my friends, I want every student understand. This is what? A sieve tube element membrane. All the students look into the picture, friends. This is what? Sucrose. Sorry, this is the sucrose. Sucrose enter near the sieve tube element membrane. But friends, the point is very clear. Do you think sucrose can easily cross the sieve tube element plasma membrane? Do you think, is it possible? No, it is not at all possible. Sucrose never ever cross sieve tube element membrane. Then how we have to bring the sucrose inside the sieve tube element? Once the sucrose enter into the sieve tube element, then only movement occur, right? So friends, now here we will use this secondary active transport. In the yesterday class I told you na, primary active transport, secondary active transport, where we will use energy from the indirect sources. Now everyone understand how we are bringing this particular sucrose inside. So friends, on the plasma membrane of a sieve tube element, there are H plus ATPase pumps are located. What are located, friends? H plus ATPase pumps are located. I want you to pay attention towards this. By the ATP hydrolysis, which got pumped out, friends, H plus got pumped out. Friends, now you know what happening. H plus creates the gradient. What it will creates the gradient? H plus creates the gradient. Then immediately what is going to happen, friends? This H, listen to me carefully. H plus creates the gradient, and this particular H plus bringing the proton. Listen to me very carefully. This H plus bringing this particular sucrose via H plus sucrose symporter. We already know, right? Symporters are the one where two solutes are coming in one direction. So how does the sucrose entering inside the sieve tube element? As the sucrose can't cross the membrane, so it requires the help. Who is doing that particular help? H plus. How the H plus can be pumped out? First of all, very simple, by the ATP hydrolysis. By the ATP hydrolysis, H plus can be pumped out. Immediately what is happening, friends? H plus create the gradient, then H plus and sucrose enter inside. Very simple. This is the apoplastic pathway of loading. Apoplastic loading is driven by the proton motive force, PMF, is capable of creating a steep uphill concentration gradient. Correct na? H plus moved from low H concentration to the high H. Then once again it is coming from high to the low. Very simple point it is. Now next type is what, friends? Symplastic pathway of loading. We all people know very well. The moment we heard the name of a symplastic, plasmodesmata, right? The movement is not at all difficult movement, friends. It's an easy movement because there is an involvement of the plasmodesmata. All the students listen. This is the mesophyll or the bundle sheath cell. Intermediate cell means companion cell, and friends, you can see this is the sieve element. Every student pay attention, friends. Now glucose and fructose combined with each other, and they are going to give rise to what, friends? Sucrose. Friends, the question here is that how does mesophyll or bundle sheath cells start moving? How does the sucrose starts moving from the mesophyll or bundle sheath cell? How does the sucrose starts moving towards the companion cell? Friends, they starts moving towards the companion cell via plasmodesmata. Don't forget it. Now you know, whenever, every student listen, whenever the sucrose entered into the companion cell, sucrose can be converted into what, friends? Large sugars. What are the large sugars? Like raffinose and stachyose. Raffinose and stachyose are very large sugars. Now the question is very clear, friends. Do you think the raffinose and stachyose, do you think they can come back into the mesophyll or bundle sheath cell? Do you think is that possible? It's not at all possible. Why? Because, friends, they are very larger sugar. Since they are very large, they cannot diffuse back to the mesophyll cell. They never come. Instead, what is happening? The raffinose, stachyose, larger sugars from the companion cell, they are entering into the sieve tube element. How, friends? Of course, via larger plasmodesmata. Finally, they will enter. So, friends, this is what one? Apoplastic pathway of loading, symplastic pathway of loading. Now the most important, right? Every student pay attention. This is the most important diagram. I want every one of you has to focus. Much more important diagram. Right now what we studied is this. This is source, mesophyll, companion, sieve tube element, nothing but phloem, and this is the xylem. We all people know that. Right now whatever we studied is what, friends? We studied right now is the loading. So what got loaded? From the mesophyll cell, sugars are loaded. From where to where? From the mesophyll to companion, companion to sieve tube element. Right now, friends, now what we people has to study. Everyone listen. This is what we have to study now. How sugars are moving. Listen my words carefully. How sugars are moving within the sieve tube element. How does that movement can be mediated? Everyone listen. That movement can be mediated by the pressure. That's why we call it this as the pressure flow mechanism. In a simple way, friends, the sugars starts moving from the source to the sink. Once again listen. Sugars are moving from the source to the sink via pressure. That's why we can call it pressure flow mechanism. Since it is explained by a scientist called as a plant physiologist, Ernest Munch. That's why we can also call it as a Munch hypothesis or bulk flow hypothesis. Any names are same. Now let me explain you elaboratively. Just pay attention. Pay utmost attention. Friends, you can see finally sugars are entered into the sieve tube element. Correct na? Of course. Friends, once the sugars are entered into the sieve tube element, you know what happens? From the xylem, which is coming, you can see, friends, the arrow is indicating now which is coming from the sieve xylem, water is coming. Now you can ask me, sir, why the water is coming? See, sucrose is what, friends? It's a solid molecule. Once again, all of you use the brain. Sucrose is what molecule? Solid molecule. Solid molecule cannot transport very easily, friends. So we have to create an environment which is a kind of liquid environment. By that it can easily move For that reason, water molecules starts entering from the xylem into the sieve tube element of the phloem. Now this is the unique statement. Now water molecule combined with the sugar. Friends, when the water molecule combined with the sugar, you know what do we can call it as? Yeah. One second, what I forgot this. Yeah, listen. I'm sorry. Once the water molecules comes inside, what will happen? Here a lot of sugars are there, right? And as I told you that sugars are solid in nature. Now the sugars has to be moved. So friends, all the students listen. Now this sugars combined with the water molecule. Once the sugars combined with the water molecule, they can create a liquid environment. Now every one of you listen. Now this liquid can easily starts coming down. Okay, the question is very clear. How they will come down? If they want to come down, there should be a pressure. So friends, how does that particular pressure can be created? Of course by the water only. The water molecule creating a great pressure on the cell wall of sieve element. Once again I want you to remember, the water molecule creating a great pressure on the cell wall of the sieve element. What do we call that particular pressure? That pressure only we can call the turgor pressure. Because of that particular turgor pressure only, friends, sap start moving. What do you mean by the sap? Water and sugar together we call the sap. What we can call the sap. Now everyone listen to me carefully. Because water creating a turgor pressure, now the sap moving down, you can see here all the sugar and water easily comes down. Okay, where they have reached now? They have reached to the sink. Now friends, when it come down near the sink, there will be a no pressure. Obviously friends, whenever there is a no pressure, water molecules starts moving back from the sieve tube elements of the phloem back to the xylem. Such a simple step it is. Then what about the sugar molecules? We will read in the next slide. So from the sieve tube element of the phloem, they will unloaded into the sink. Friends, one more interesting point is what here? If they want to unload into the sink, once again what they required? They compulsory require the companion cell. So friends, I will re-explain once again. I want whomever did not understand, pay attention. What is happening first? From the mesophyll cell, sugar enter into the companion to the, it entered into the sieve tube element of the phloem, which we can call loading by the symplastic pathway, apoplastic pathway. Now what is happening, friends? This particular water molecules entered from the xylem into the sieve tube element of the phloem. It create a great pressure, create a turgor pressure. Because of that turgor pressure, water molecule and sugar together, what we can call sap. The sap starts moving down. Till which area they will move down? Till it reach the sink tissues. What are sink? I told you na. You can call stem is the sink. You can call root is the stink. You can call tubers or the stink. A sink, any region will be the sink which cannot produce the sugar. Which cannot produce the sugar on their own. We can call sink. Once we come down, there is no much pressure, friends. That's why once again water molecule will go back from where to where? From phloem to xylem. Now let's try to understand how does the unloading occur. See, unloading, whatever the statements I will highlight here, just remember that. We discussed phloem loading also, symplastic pathway, apoplastic pathway, I told you. What I want you people to remember, phloem unloading, just remember these 2 points enough. If the sugars wants to unload into the stem or the shoot, they will always use the symplastic pathway of the unloading. Always. But if the sugars wants to be unloaded into the rest of the plant body, the entire part of the plant body, whether it may be root, whether it may be tuber, doesn't matter, it will always follow the apoplastic pathway. Friends, if you remember these 2 points, that's more than sufficient. If the sugars wants to be unloaded into the stem, they will use the symplastic pathway by using plasmodesmata. But apart from the stem, if the sugar wants to be unload into any part of the plant body, any part apart from stem, either root or tuber, what kind of pathway it will follow, friends? It will basically follow the apoplastic pathway. Always remember that. So friends, that ends this particular transport in plants. So what are the various topics that we are left here? We are left with the sensory photobiology, very rigid topics, but important topics, friends. Then followed by the stress physiology chapter. Okay, so it will take another 3 classes, friends, because stress physiology is a kind of a very rigid topic, friends. We have to read it, very rigid it is. Sensory photobiology is somewhat interesting because we can read some experimental based concept, but stress physiology is purely theoretical and very dry concept, but we have to read without fail. Okay, good morning Tanmay. Okay friends, now everyone answer which chapter you want me to start. See, maximum in 3 days this plant physiology will be done, right? Anyway, in the Unacademy I am doing the unit 7. Unit 7 also will be done by 13th or 14th. Let's take it will take 15th. Now tell me, you want me to start in the YouTube unit 1, or you want me to start unit 4, or you want me to start unit 5? You choose any one unit. Once the human physiology is over, I will start the second unit very immediately. Tell me friends, which unit you want me to choose? Majority of people, whatever they say, I will go with that. Majority. Even I'll ask in the group also. Friends, unit 1 is the very large unit. Unit 4 is very larger than the unit 1. Okay, unit 4 is also very big unit. Okay, you people decide, friends. I will raise a poll in the group also. Based on your all opinion, I will go with that particular unit only. Okay. Okay friends, that's all for now. In the tomorrow morning class, I will finish this, what is that called as? I will finish this sensory photobiology chapter. Okay, we will discuss what chapter, based on your all decision only I will take that particular unit based on the majority. Okay, okay, done. Okay friends, I will see you at the 8:30 AM class in the Unacademy, where we will be continuing the respiratory system. Okay friends, thank you all of you. I'll see you at 8:30 AM sharp. Bye bye. Okay, which unit we will decide? We will decide in the Telegram group.